Hi everyone, my name is Madhu and today I will be presenting on the behalf of Purdue iGEM, a competitive synthetic biology research organization, and I will be presenting our 2020 project, Argo, a novel Argonaute-mediated microfluidic COVID-19 diagnostic device. Throughout 2020, COVID-19 has affected lives on a global scale, resulting in over 1.31 million deaths globally. The pandemic put the capacity of institutions and companies' production of rapid viral diagnostics to trial. Current tests related to COVID-19 have been developed in the form of a nasal swab test, saliva test, and antibody test. Of these tests, the two diagnostic ones can take between 1 to 14 days to receive results. Due to governmental pressure and the need for rapid testing, many of these rapid tests were accelerated through the regulatory approval process, resulting in compromised accuracy. Current tests in the market have false negative rates around 25%, meaning up to a quarter of those who test negative may be infected. This, along with the 1 to 14-day test runtime, results in the risk of transmission of the disease, as depicted by the transmission diagram. Thus, there is much to be improved within viral diagnostics. In efforts to improve upon viral diagnostics, Purdue iGEM decided to research how Argo, or Thermus thermophilus Argonaute proteins, and microfluidic principles can be utilized to develop a rapid, reliable, and affordable viral diagnostic device. Our solution was built upon three main principles: reliability, rapid testing, and affordability. To ensure a solution embodied these three principles, our research was divided into two sections: hardware and biologics. The first step was to design the biological system of our device. The science behind our system can be summarized by the following steps. First, a sample of patient saliva is inputted into the microfluidic chip. Viral RNA from the saliva is extracted by an embedded chitosan capillary to be separated from the remainder of the sample. Next, RT-RPA reagents are inserted. These reagents and the RNA continue through the device to the reaction chamber. Here, the RNA is converted to DNA and it is amplified to produce a large amount of DNA via RT-RPA, an isothermal process similar to RT-PCR. Then, Argonaute's molecular beacons enter the device. Argonauts cut the DNA to generate fragments of single-stranded DNA. These DNA fragments bind to molecular beacons, causing them to hybridize and produce fluorescence. Finally, this fluorescence is quantified via a cell phone spectrophotometer to interpret results, which are outputted on an app. As shown in the previous video, our device has four main biological processes to tackle: a chitosan-treated capillary, which purifies RNA in the saliva sample; RT-RPA, which converts RNA to DNA and amplifies target DNA sequences; an Argonaute-mediated system, which cleaves DNA with base pair specificity; and molecular beacons, which produce fluorescence in response to the presence of a complementary single-stranded DNA fragment. The first component of the system is the chitosan capillary. Chitosan is a bioadhesive polymer that binds either positive or negatively charged molecules depending on the pH of its environment. The chitosan captures a lysed viral RNA in acidic conditions and releases the RNA in basic conditions. This pH-based capture, purification, and release system is illustrated in the diagram. By using the system, we can potentially extract high concentrations of RNA from saliva in the device itself at speeds as fast as 90 seconds. Once the RNA is extracted, it needs to be detected by our Argonaute protein. Argonaute is sensitive to double-stranded DNA, thus we need to convert the extracted RNA into double-stranded DNA. Via extensive literature search, we decided to use RT-RPA to amplify the target RNA into double-stranded DNA. RPA is a novel isothermal DNA amplification method and was chosen for Argo as literature demonstrated its ability to provide detectable concentrations of DNA within 10 minutes in a microfluidic device. With the addition of RT, or reverse transcriptase, to RPA, we're able to convert the RNA into DNA for further DNA amplification. Now that our viral RNA is converted to double-stranded DNA, the Argonaute system can be utilized for detection. Argo, or Thermus thermophilus Argonaute, is a member of a unique class of endonucleases called Argonaute. The Argonaute proteins uptake designed DNA guides during incubation and use the guides to cleave a specific target region of DNA. As seen in the diagram, this property is used to produce single-stranded DNA fragments specifically from COVID-19 DNA. These single-stranded fragments then can be tagged or quantified using molecular beacons. Molecular beacons consist of a 5' fluorophore quencher, a probe sequence flanked by 2 stems, and a 3' fluorophore. The beacon folds spontaneously as a hairpin structure that quenches fluorescence. Once the single-stranded DNA fragment produced from the previous Argonaute cleavage binds to the target sequence, the molecular beacon undergoes a conformational change, separating the fluorophore from the quencher and emitting a fluorescent signal that is detectable via spectrophotometry. Thus, if a significant deviation from background fluorescence is detected, the COVID-19 genome is present in the sample and the patient is COVID positive. To design all biological parts of the system, several team members collaborated on a Python script that interfaced with online oligo design tools. This program was used to unify and facilitate the interdependent design process for RPA primers, Argonaute DNA guides, and molecular beacon. The program, upon inputting a host genome, suggests optimal TTAGO DNA guides, RPA primers, and potential molecular beacon sequences. The parameters for optimization were found through a well-documented iterative design process. Results of the code optimizations can be seen in these images. The chitosan capillary adsorption was also modeled to determine the optimal capillary length for max RNA adsorption. We developed a model of RNA absorption by chitosan using a Lang-Mer isotherm absorption model. This model assumes only one layer of RNA can bind to the chitosan capillary and that there are no interactions between adjacent RNA molecules. Upon thermodynamic derivation of the absorption and desorption reactions of RNA by chitosan, it was determined that the amount of absorbed RNA to be a function of the maximum amount of RNA absorbed and of the equilibrium constant of the absorption-desorption reaction with chitosan. Both constants were experimentally determined and readily available for an 8 cm capillary in current literature. Using various equilibrium constants and assumptions regarding the model, we plotted our existing model as a function of RNA elution with varying lengths. As a result, we produced a three-dimensional graph that shows the estimated RNA elution amount as a function of capillary length and equilibrium concentration. In addition to the biological components, our team worked on developing a model for the physical prototype of our device. Through iterative design process, we produced two models, one of which was deemed the final model after consulting with academic and industry experts on the design. Our final model consisted of a 25 mm by 25 mm polydimethylsiloxane PDMS microfluidic chip attached to a set of syringe pumps for fluid actuation. The final chip design was optimized by minimizing dead space via our small dimensions and initiating mixing through simple acoustic vibrations. The RPA reaction chamber indicated in blue was also modified to contain an outlet on the topmost layer to account for vapor pressure buildup from the RPA and Argonaute reactions. We also allocated space for a control reaction on our chip to make sure our device was compatible with regulatory device standards. To accompany our microfluidic chip, we also designed a small portable metallic heater to be placed underneath the reaction chamber of our chip for reaching the optimal temperatures for the RPA and Argonaute reactions. The heater uses a proportional integral derivative control algorithm capable of maintaining temperatures from 30 to 90 degrees Celsius with 1 degree Celsius accuracy and was also suitable for point-of-care applications. Through our research project, we derived the following results: a finalized CAD model of our microfluidic diagnostic device and a finalized heating device to accompany it, the determination and design of all the biologics in our system, which include RPA primers, TTAGO guide DNAs, and the final molecular beacon design. We were also able to model the effects of the chitosan capillary length on RNA absorption to determine the optimal capillary length for our system. Overall, the theoretical design of our diagnostic device met our standard for a rapid, reliable, and affordable viral diagnostic device. We also were able to produce a 20-minute formal presentation, poster, and wiki website as deliverables for the world's largest synthetic biology competition hosted by IGEM called Giant Jamboree. Based on our results, we determined that we can in fact develop a rapid, reliable, and affordable COVID-19 viral diagnostic device using Argonaute proteins and microfluidic principles. We also determined that our device can easily be adapted for the detection of other viral diseases and even multiplex viral detection in a single sample. Thank you for listening to Purdue IGEM's 2020 project presentation. We hope you are staying safe and having a good day.